I should say, if you'd asked me a few years ago if we'd be studying infectious disease, I would have laughed, because the truth is, like many of you, we were impressed into service. I'm just a yeoman cancer immunologist. Our research program has historically been focused on engineering immunoreceptors and in their ligands for cancer immunotherapy. But it just so happened that as the pandemic was hitting, we were shifting our research focus, or rather adding a research focus in studying and understanding patient autoantibodies. And so we were really primed to study this question. Before I get started, though, I just want to acknowledge that our research is based on protein engineering and creating pharmacologic tools. And some of those tools have been commercialized. And I've been involved in the efforts to bring them into the clinic, as well as to advance those tools, some of which I'm talking about today. And so these are my competing interests. So as I was saying, we've long had an interest in using immunopharmacology to probe and understand the immune system. Over the past few years, though, we've been interested in the converse of that question, trying to understand how the natural pharmacology of the immune system, that is to say, patient autoantibodies, can have a big impact on disease outcomes. The idea here being that, of course, when you think about autoantibodies, you think about their central role in mediating rheumatologic diseases, autoimmunity, where you have antibodies that target tissues and other aspects of the body and drive damage. But there's a growing body of evidence across many different types of diseases, nearly every facet of disease, that some antibodies are actually protective. For example, in autoimmune disease, patients who make autoantibodies against cytokines have less severe disease. In cancer, patients who make antibodies against their tumors live longer than those that don't. And even in neurodegeneration diseases that aren't typically thought to have a central immune component, antibodies have been found against proteins like amyloid or prions or tau, alpha-synuclein, that appear to ameliorate the disease. So we're really thinking a lot about autoantibodies as independent risk factors, modifiable risk factors for disease, and thinking about antibodies almost like genetic alleles. And so we've been really intrigued by this question. Well, how do we find these really functional central disease-modifying autoantibodies in an unbiased fashion? All those examples I just gave you were kind of obvious targets, given what we already knew about the pathophysiology of those diseases. Why did we study HER2 in breast cancer? I think it's pretty obvious. It's it was a known driver oncogene, proto-oncogene. Why would we look at cytokines in autoimmune disease? Again, a pretty straightforward hypothesis. How could we be unbiased? And in thinking about the antibodies that we're most interested in, of course, we're biased. We're immune receptorologists in my lab. We posited that the most interesting antibodies that would influence host physiology would be those that recognize extracellular proteins in the extracellular compartment. And the rationale is really straightforward. Antibodies themselves are large secreted proteins, and by and large, they're going to come into contact with antigens in the same compartment. Certainly they can bind, and they do bind, proteins that are cytoplasmic, nuclear, et cetera, and they have an important role in promoting pathology, for instance, due to immune complexes that can form when cells are lysed, et cetera, as has been well characterized. But by binding proteins that are extracellular, they can actually influence physiology, normally. So, antibodies could, for example, inhibit or enhance, receptor ligand interactions. They could directly agonize or inhibit receptors, or they could mark different cells and proteins for destruction, through antibody effector function. So the idea here is that these extracellular targets would be the most impactful. The problem is that they're actually also the most difficult to study from this perspective. In seroreactivities against the extracellular proteome, the exoproteome has long been a big gap in our understanding of autoantibodies. And the main reason is this: most antibodies in general, most autoantibodies recognize proteins in their three-dimensional folded conformation, as opposed to linear epitopes that are resistant to denaturation. But the problem is that these extracellular proteins are among the most fragile. They have unique folding requirements. They have posttranslational modifications like disulfide bond formation, glycosylation, signal peptide removal. And it's long been appreciated that they're tougher to work with. And so unfortunately, these types of antigens are not well captured by conventional approaches like protein and peptide microarrays, or peptide display systems like on phage, very innovative phage platforms like FIPSeq or bacterial display. And so this is really where we set up STEP. They said, well, this seems like a problem, A, that we're interested in, B, that we think that we can solve for, given the types of technologies we have, in our lab. And in one idea we had is, well, it turns out the system that we predominantly use to engineer proteins, yeast surface display, we use it for directed evolution, could be repurposed for extracellular autoantibody discovery. And so we made a big bet that most extracellular and secreted proteins could be properly displayed on the surface of yeast. And so we've, over the last few years, made a very large library of several thousand extracellular and secreted autoantigens tethered to the surface of yeast, the yeast cell wall, such that this library is fairly comprehensive, and each protein has its own set of barcodes that we can use to track them. And so what we've done is we've taken this library and we've developed an approach that we call REAP, Rapid Exoproteome Antigen Profiling, or extracellular antigen profiling, REAP. And it's a pretty straightforward technology. What you do is you take a bit of patient sample, you mix it together with the yeast library, and then you simply isolate the yeasts that are coded in patient IgG using magnetic separation and sequence the barcodes. So you turn an antibody antigen binding event into a sequencing readout. And so initially, and this is what we've published on so far, our library had about 3,000 of these proteins. We've since expanded it to about 6,000. But what I'll present to you today is work from the smaller library. But it's actually fairly robust. It only requires a small amount of patient sample, and it allows us to multiplex together hundreds to thousands of samples at a time. Just given the fact that next generation sequencing is so high throughput and really lends itself to multiplexing. So like I said, I promise the setup for the technology is worth it, given what we found in COVID, but I just want to give you a little bit of color about the performance of this assay that we had been developing right as the pandemic hit. And so we first applied it to a setting where we knew that important extracellular autoantibodies should be present. And so we decided to study this disease called APS1 or APOSED. This is a disease, a rare genetic autoimmune disease caused by loss of the AIR genes. They lack central tolerance. And as a result, they get devastating autoimmune polyendocrinopathies, antibodies against different endocrine tissues, but they also paradoxically get immunosuppression because they make extremely high affinity, high titer antibodies to a range of different cytokines like the type I interferons, IL-17, IL-22, et cetera. And so we thought, this could be a great disease to really benchmark how well our system is working. Can we detect these known knowns, if you will? And so we obtained almost 80 of these samples from a collaborator, Mihalis Leonakis, and profiled them with REAP. And what you're looking at here in the heat map on the left, is basically a summary of all the autoreactivities that we found in any of the patients or healthy control. And as you can see, each patient here is a column. Each row is an autoantigen for which we found at least one individual having an autoantibody. But actually, if the matrix here, the number of rows should extend to 3,000, and yet, we only detected, a few hundred antigens. Sorry about that. So, it's a pretty sparse data matrix. But two is actually that we found, by and large, overwhelmingly, the predominance of those known knowns, those anti-type I interferon autoantibodies that are nearly universally present in this condition, antibodies against IL-22, again, nearly universally present. We found the expected frequencies of antibodies against IL-17, type III interferons, IL-5, et cetera. So these are all sort of the known knowns. We found other tissue autoantigens like gastric intrinsic factor. This is associated with vitamin B12 deficiency in these and other patients. Lipocalins, again, known autoreactivities. Then we found a bunch of completely novel, previously undescribed autoantibodies against a wide range of different antigens. Some of them were relatively private. Others were somewhat public, like this autoantibodies against this glycoprotein hormone, GPHB5, and it was associated with patients who had hypoparathyroid complications of their disease. So again, we were pretty impressed by the performance of the technique to detect antibodies that we knew should be present and to find new ones in this well-characterized patient population. The other thing we found, though, was that REAP is not just a yes/no answer. That the enrichment that we see in the assay, the more yeast you're pulling down, that actually corresponds with higher titers of antibodies. If you titrate the amount of patient IgG, you get a sigmoidal dose response curve, and the relative rank order of these curves matches up well with what we see with ELISA. So you can essentially calculate like virtual titers with this approach. Okay, so what about COVID? So as I was saying, we'd just gotten this technique off the ground as the pandemic hit, and so early on we were sort of primed, no pun intended, to think about autoantibodies in this condition, and certainly there were some clues early on that they could be important. So in the original SARS-1, there were reports that autoantibodies could be found against, for instance, the lung parenchymal tissue. In patients who were severely impacted by SARS-1. And there have been some implication that perhaps there was molecular mimicry involved, although that wasn't proven. And then there were reports profiling patients with COVID-19, it was appreciated that they did have really peculiar activation of pathological B cell responses, like these extrafollicular double negative B cells, as described by Ignacio Sanz's group, that had previously been characterized in lupus. So even relatively early on, their B cell compartment appeared to be problematic. And then of course there were numerous reports showing elevation of conventional autoantibodies against nuclear antigens, phospholipid antibodies that could contribute to the coagulopathies those patients have, and other conventional rheumatologic antigens. And then, really, I would say, one of the most important reports of the entire pandemic was out of Jean-Laurent Casanova's lab, where they found not antibodies that were generated by COVID, but antibodies that greatly impacted COVID, which is the implication of these anti-type 1 interferon autoantibodies that were heavily enriched in patients with severe disease that neutralized the interferon, and presumably given the central role of interferon in protecting patients, that those patients be predisposed to a particularly severe infection. That work has been since widely replicated by our group, by others, and expanded by the Casanova group. Now profiling 30,000 different patients for the presence of these antibodies. So this is a really important study. I'll talk more about it. So again, really convergence of evidence that not only can COVID drive the production of autoantibodies, at least some clues for that, but it also can be impacted by the presence of autoantibodies that are influencing immune responses. So that was sort of the setup. So it turns out that at Yale, my colleague Akiko Iwasaki, Albert Ko, Saad Omar, they had very, they had, in the early stages of the pandemic, they had the foresight that we would need to establish, really robust, well-characterized, well-phenotyped cohort of patients infected with the coronavirus to understand the impacts, across, essentially to do the translational studies. That's called the impact cohort. I think the results stand on, this has been just a tremendously successful, important, influential cohort that's been cited in dozens of studies at this point. And so we were really fortunate to get access to that cohort of over 200 patients seen in the Yale hospital system, mostly hospitalized patients. So a bit of a skew toward severely affected patients, but really great clinical phenotyping, immunological phenotyping, and longitudinal collections over the course of their hospitalization. And so we took these approximately 200 patients, profiled them by REAP, and what we found to us was pretty eye-opening. At first, we found that COVID patients had elevated levels of autoantibodies globally across the board, and that the patients who were the sickest, they had the most total numbers of reactivities. In fact, COVID patients in general had higher levels of autoantibodies than patients with severe lupus. And some of them actually had levels of antibodies that approached what we saw in APAsets. So really quite striking just how autoreactive these patients were. Another thing that immediately jumped out at us too is that many of these autoantibodies were directed toward the immune system itself. So for example, we found as the Casanova group did that yeah, there were a number of patients who had these anti-type 1 interferon autoantibodies, and notably they were clustered in this severely affected cohort of patients. So here we've ordered the patients according to their severity. Here is the most severe critically affected patients. These are more moderate, asymptomatic, and mild, and then on the far right are healthy control. So definitely found interferons, but really that was just the start. We found numerous other cytokines, immune cell surface receptors, chemokines, inflammatory mediators across the board. But I just want to dive in a little bit to those anti-type 1 interferon autoantibodies. Again, the theory here being that patients who have these antibodies are unable to mount the needed interferon response to control the virus. And so again, we confirmed that indeed these antibodies are present across multiple different, interferon alpha and omega subtypes in the severely affected patients, most notably. But we were able to extend this and really show that was associated with impaired virological responses to the virus. So these patients had higher levels of virus at initial onset and when they initially presented to the hospital. And they, as you can see, they also had much slower clearance of the virus over the duration of their hospitalization compared to patients who didn't have those antibodies. So really, we thought this is an important sanity check for our technique in our cohort, but also we were able to extend and provide further evidence that these interferon antibodies are really impacting patient outcomes. But like I said, interferons were really just the tip of the iceberg. We found over 30 different cytokines, 24 chemokine pathways that were targeted by patient autoantibodies in COVID. Really diverse sets of targets, not just the interferons, but IL-1 family member, colony stimulating factors, type 2 cytokines, TNF superfamily member, et cetera. They're present in fairly decent titers, 1 to 100, 1 to 10,000 range. And importantly, too, they were functional. So here's an example of a patient who had antibodies against, GM-CSF, and if their serum, specifically neutralized GM-CSF signaling compared to, patients who didn't have those antibodies. We also found patients with, anti-chemokine antibodies had impaired, a GPCR signaling downstream of those chemokines as well. So these were largely functional antibodies. The issue here, though, is that the allele frequency, if you were going to compare these to genes, it really varied widely. And unlike the interferons, where we could kind of bootstrap our way to function by just seeing them in so many different patients, for many of these cytokine pathways, we only saw them in a handful of patients, not really enough to by itself drive conviction that they were impacting the disease. So what we did is we tried to model preclinically the effect of these cytokines in mouse models of coronavirus infection. And so my colleague Akiko Osaki-san, they used the K18 ACE2 mouse model where they could infect this mouse with coronavirus, and then we would pre-inject those mice with antibodies meant to mimic the different cytokine responses we found. Like, for example, if you inhibit the interferon pathway, you can see that the mice do a lot worse, as expected, consistent with what we saw in patients. But we also saw that other cytokine pathways had similar impacts, like IL-18. Again, blocking IL-18 was profoundly, produced profound bad outcomes in these mice. IL-1 beta, IL-21, GM-CSF, and this allowed us to sort of, gain more conviction that these responses we saw in patients, could be influencing immunological function in the patients. But it wasn't just immune signaling molecules that we saw in patients. We also saw antibodies that targeted the surface proteome of different immune cell populations. So for example, we found many patients who had antibodies against B cells, paradoxically pretty interesting. Although not that uncommon, this is also seen in other autoimmune diseases. B cells are thought to be a particular, a target of autoantibodies, just given that they're present in the inflammatory milieu. Those antigens on B cells are nearby, where these incipient antibody responses are forming. But I'll give you some examples. We found a patient who had extraordinarily high titers of autoantibodies against CD38. And those antibodies drove really profound, ADCP, phagocytosis of B cells ex vivo. And when you look in the blood of these patients who have these, B cell directed antibodies, they have lower levels of B cells, as you may expect, consistent with depletion. And as a result, they also show impaired humoral responses to the virus. If you look at their spike responses, they have a lot lower levels. So again, this really paints a picture that these antibodies potentially, as is seen with iatrogenic B cell depletion in patients who get, say, like anti-CD20 antibodies, really drives worse outcomes by dampening humoral responses to the virus. The issue here is that it can happen endogenously. But it wasn't just B cells. We saw similar patterns with monocytes. Even an example of an individual patient who had anti-CD3 epsilon antibodies, who had very severe disease and had specific depletions of CD3 positive cells like CD4, CD8, and NKT, but not other lymphocyte populations like NK cells or B cells. And those antibodies also drove, antibody-directed, effector functions ex vivo. So certainly the immune system was impacted by these autoantibodies, and that seemed to potentially influence, immunological, virological outcomes in the patients. But I just want to point out, we found a lot of autoantibodies against tissue autoantigens as well. Notably, a lot of autoantibodies against the central nervous system, vascular and blood, express proteins, platelets, coagulation factors, connective tissue, et cetera. And because there were just so many different antibodies and the clinical phenotypes of these patients were so varied, it was tough to directly compare or deduce particular autoantibody responses in specific clinical phenotypes in most cases. These patients had a lot of reasons to be sick, but we were able to draw some inferences. So for example, we found that patients who had antibodies against the orexin-2 receptor, hypocretin-2 receptor, they skewed toward having worse neurological function, crudely Glasgow Coma Scale here, which is, admittedly, not the best metric, not an ideal metric, but, a pretty striking correlation of antibody reactivity and neurological function here. And these antibodies were indeed functional. They inhibited the activity of this pathway when you look ex vivo. But of course, this really, drives the question, if these autoantibodies, develop during COVID, and certainly we have evidence that that antibodies.
Hey, Aaron, sorry to interrupt. Yep. This is a 2-minute warning.
Yep, I'm on my last slide here. That certainly there were examples of antibodies that likely predated the infection. They were present before the anti-spike responses came up. But we also saw examples of antibodies that were not present when patients first came into the hospital and then were acquired during their hospitalization, very clearly being tied to infection. And that really drives the question of, could these antibodies underlie some of the long-term impacts of infection, like the post-acute sequelae of COVID, long COVID? And that's an active question that we're looking at in our lab. And in fact, we've now looked at some of these patients that we initially studied in the acute setting and are following up with them, some now, a year out from infection, to see, do some of these antibodies continue to exist, or have they waned? In some cases, we see they wane quickly. But I will say, we're currently working on a story. It's not quite yet ready to present, but the news is a little bit disturbing. We have now seen some examples of patients who, over 300 days out from their infection, have maintained high levels of acquired antibodies that came up during their hospitalization. And we're now looking to see, are those antibodies associated with the development of long COVID syndromes? So just to wrap up, hopefully I've been able to convey that this approach we've taken with REAP has allowed us to detect these functional autoantibody responses, not just autoimmune diseases, but in COVID, that the autoantibodies we found could impact immune function and virological control. And again, this leads us to the hypothesis that these autoantibodies could underlie a lot of the varied clinical phenotypes that are seen, not just acutely, but long-term post-COVID. And we're looking at this not just in long COVID, but also in patients who are individuals who are being vaccinated or have vaccine-related adverse events. In a sentence, I'll just tell you the news is good. We're not seeing autoantibodies come up with vaccination. We actually really don't even see autoantibodies in some of these adverse events like myocarditis. And of course, we're really looking to extend our studies with the REAP system to a wide variety of diseases. I'd just like to briefly acknowledge the folks who did this work. A trio of really talented students in my lab: Eric Wong, Ela Dai, Jill Jaycox, our stalwart collaborator, Akiko Asaki in her lab, as well as the Yale Impact team in our funding. Thank you for your attention.
Well, thank you, Aaron, for that super interesting talk. Again, I'll remind people if they have questions to please raise their hands. And I'd like to start off with just some very perhaps naive questions. If you just do a survey of the general population, do you find autoantibodies in lots of individuals that don't have any particular disease phenotype?
Absolutely, yeah. If you look at healthy individuals, most of us have a smattering of autoreactivities, and that makes you wonder, okay, are they just not functional? They're not relevant, or it also raises the question that those autoantibodies could be in some ways protective, right? And I'll give you an example of that. So a lot of these patients walking around with anti-type 1 interferon autoantibodies, they don't know they have them. And in fact, there's some reports that those autoantibodies are protective against diseases like lupus. But it's a two-edged sword, right? In that those same autoantibodies, predispose you to bad outcomes in COVID. So, like I said, we're really thinking about autoantibodies as alleles, as disease modifiers, and I think it's important, like your question gets to, not just to look in disease states, but to do a really broad survey, of the healthy, normal, and with really well phenotyped, control patients.
Perfect. I think Zhizhang Sheng had her hand up, his hand up first. So
can you hear me?
Should be able to talk. Yes.
Sure. So very nice talk. Have you ever isolate those kind of autoreactive B cells and see how they arise in the COVID-19 patients, like why they are arising? Are they possibly being first target specific SARS-CoV-2 antigens and then also carrying autoreactivity or other scenarios here?
We haven't in my lab, but Ignacio Sanz's group has a preprint where they've done exactly that, and they've looked at these antibody secreting cells in the blood of COVID patients and phenotype them. A lot of them are coming from the extrafollicular response, these double -2 cells, and they've now cloned out some of those antibodies and surveyed their reactivity to COVID antigens, as well as autoreactivity. And what's interesting is that a lot of the repertoires being used by those cells are this VH434 framework that is predisposed toward autoreactivity or polyreactivity due to a hydrophobic patch there. And it turns out a lot of these, what they found in this paper is a lot of those B cells have not mutated. They're near germline-like sequences. They're just sort of like a loss of peripheral tolerance, and that may explain why some of these responses are coming up. I don't know yet if those particular B cell subsets explain all of the autoreactivity. I actually doubt that they do, because that autoreactivity tends to be more transient, whereas we're seeing some responses wane, but other responses remain stable. So likely there are multiple pathways of autoantibody production.
I agree. I think the VH4-34 genes antibodies, usually the germline antibodies are autoreactive, but most of them are being depleted during B cell maturation. But it's the first time I haven't heard that those kind of antibodies are reactive to like cytokines. Those kind of is very interesting to look into.
Yeah, check out that preprint by the Sans group. It's very interesting.
Right. Thank you.
Okay, and Ali has a question. You should be able to.
We have time.
Yes, go ahead.
So, nice talk, Aaron. One of the things that I was wondering about listening to your talk is that can you actually look in specifically in people with very specific defined autoimmune diseases, and with at least known targets, like there are many autoimmune diseases where we know actually the self proteins that they are having cross-reactive or antibodies against. So, and then in a way, can we define a threshold where you have, if you're using your method, where we have that level of antibody is associated with the disease, and then probably after defining that threshold with your method, then going back and start looking at those COVID, severe COVID, or actually severe other viral infections. There are many others, and I can tell you we have a few samples if you're interested for other viral infections. But it will be, I think, I know that the clinical outcome is probably going to be hard, as you mentioned, but probably establishing that threshold based on those diseases will be great.
Yeah, I really appreciate the comment here. And in fact, one of our biggest goals of our research and why I started this company, Seronova, to really bring in a large amount of resources, is to create a huge serological atlas of tens of thousands or more patients to really understand broadly in a non-hypothesis based way, what are the frequency of these antibodies that we think we know? What are the ones we don't know, right? And look at them, across many different diseases, not just in, a targeted hypothesis based way, and really see how they influence patient disease outcomes. I would love to collaborate with you on those other infectious diseases. A question we often get is this COVID specific? The answer is probably not. It's probably not COVID specific, but I think it's more common in COVID than other infections, potentially because there could be some innate reactivity of some of our germline frameworks to COVID antigens, which is kind of interesting. I don't think there's molecular mimicry going on, but what may be happening is that COVID could be tickling some of these polyreactive cells like the VH434 more so than other pathogens. But yeah, no, again, we're looking to apply this broadly and take a 360-degree view. Really briefly, I will say we have benchmarked REAP, on sort of known knowns, like for example, in myasthenia gravis, we can find, MuSK, for example, but we don't find everything. Some antigens don't display properly on REAP. So you take what you can get, right? But overall, it's okay. It's pretty good. It's not perfect.